Welcome, everyone. Good evening. Thank you very much for joining us tonight. I'm Julia Hanmer. I'm BSBI's Chief Executive, and it's great to be here with you tonight for our webinar on getting started with flower anatomy. It's part of our Northern Ireland Botanical Skills project, which is supporting botanists with training, events, and recording, so that we have more people and more data to inform nature recovery. And a huge thank you to our funder, the Department of Agriculture, Environment and Rural Affairs in Northern Ireland, who've made this project possible and the webinar series possible. So tonight is the second in a programme of talks that we're running on Tuesday evenings through the winter from through till February. The details of all the talks are on the BSBI events page. All the talks are being recorded and will be uploaded to our YouTube channel afterwards. If you enjoy this talk, do consider joining BSBI. If you join us now in November, you get 14 months of membership for the price of 12. So it's a real special offer time of year. There will be a Q&A at the end of this talk, and feel free to put questions in the Q&A box. So have a look on the Zoom bar for the Q&A box throughout the talk when they occur to you. Or at the end of the talk, and we will get through as many questions as we can at the end of Jen's talk, at the end of the evening. So I'm delighted to introduce Jen Farrar. Jen works at BSBI as Botanical Skills Officer, Northern Ireland. Jen's been involved in teaching plant identification and plant and flower morphology for over 15 years. She teaches botanists from complete beginners to those that are studying for masters in plants taxonomy and biodiversity, which she's done in the past for the Royal Botanic Gardens in Edinburgh. And in her current role, Jen is particularly keen to demystify looking at plants and assist people in building confidence in their observational skills and identification skills. So thank you, Jen, and over to you.
Good evening, everybody, and thank you so much for coming along to this webinar. It's really nice to have you here. I would like to start off by just saying that this evening I'm going to be looking at flower anatomy and sort of how the different types of anatomy we see in flowers and some of our fruit has come about over time. Sorry, just having a moment. So what we will cover this evening are the basic organs of a flower, their position relative to each other and how that is patterned. How to make a summary of the number of organs you see and a potted evolution of flowers, and then changes in flower characteristics, especially in relation to the co-evolution of flowers with insects. So all flowers have reproductive organs. It's important to remember that things like sepals and petals are not actually necessary for a plant to reproduce. All it really needs is male reproductive organs and female reproductive organs. So even the most reduced of flowers, like you would see in a grass, have reproductive organs. They have proper flowers. They have male and female parts together. The difference, however, whenever it comes to something like a conifer reproduction, the flowers are in particular form. So the male reproductive organs have particular anatomy, and the female reproductive organs have a particular anatomy as well. Whereas in conifers, you have male cones and female cones, and the female cones take a long time to, are quite woody and take a long time to mature, and the male cones produce masses of pollen and then drop off. So our male reproductive organs are our stamen, and the stamen is made up of the filament, which is can be a very long piece of tissue that runs from the very base of the plant, the base of the flower, right up into sort of the open air, if you like. And at the top of that, we have what are called anthers. And these are where the pollen is produced and develops and matures and then is released from. These can be absolutely huge. They can be tiny. They're not always yellow. Sometimes you will have sort of orange, purple, blue pollen. So quite an interesting range of colors. And the stamens are male reproductive organs. So these are a necessary part of the reproduction of a flowering plant. And generally, they are together with the female reproductive organs. In terms of whenever it comes to being able to shorthand how many organs you have in your flower, I'm just going to introduce this term here. Andrecium just means the male reproductive organs. It's a collective term for that. And we will come back to that shortly. The female reproductive organs are called collectively the carpel. That's made up of the stigma, which is the very tip here, which is the receptive part of the carpel. This is where pollen lands and germinates, produces what's called a pollen tube, which travels down the style, around the edge of the ovary, and comes into contact with the ovule, fertilizes that, and then that becomes a fertile seed and develops from there on as a seed. The style is this sort of area of tissue that connects the receptive tissue with the top of the ovary. The ovary, of course, is very similar to other ovaries you're maybe aware of, in that it's what holds the ovules, so the eggs, the female part of the reproductive cycle. And very similar to, say, human eggs, these only have half of the genetic material necessary for a full plant. The other half, of course, needing to come from the pollen grain and the male gametes that are there. And the term that we need here for our shorthand way of counting our floral parts is gynecium, which again, we will come back to shortly. Sorry, seem to have mislaid my area. Thank you. Sorry about that. Flowers will often sit on a structure called the receptacle. And the receptacle is what's left of, it's the beginning point of all the organs whenever the flower is extremely early in its development. Sometimes this receptacle is joined to the stem of the plant by what's called the pedicel, but basically flower stalk is fine. And the receptacle has developed a multitude of functions as we go through the evolution of flowering plants. So it's an important area to be aware of. Then we have our, oh, I beg your pardon. We also have sepals that are one of the leafy type organs that we see protecting the developing flower. We can also have smaller scale-like structures called bracts and bracteoles. Unfortunately, those aren't shown in this particular diagram, but they are also important parts of the development of the flower in terms of protecting it from both the elements and from any herbivores that might come along. Of course, many flowers have petals. We are aware of the showy, colourful ones, the big showy, colourful ones that we all love. But they can be absent as well. They can be any size or shape or colour too. So they can be incredibly small, and they can be quite large. And the same goes for the sepals. The sepals and petals are quite late evolving in flowering plants, so they have diversified hugely as well as the actual flowering families. In monocots, the sepals and petals frequently look very alike, and then in that circumstance, they're called tepals. Tepals are basically any petal-like structure which is not easy to differentiate from those structures which would have originated as sepals. So it's easier to just call them a simple tepal. In many monocots, and I say many because whenever we get to the sedges and grasses, and also to things like gingers, the flower has been reduced and some organs have changed form and function. So the parts of the flower will no longer be in orders of 3, even if that is the ancestral route. So for this wonderful bog asphodel, we have 6 tepals, and we also have 6 of these wonderfully fuzzy anthers and filaments and anthers with this fantastic orange pollen. If we have a look at the gynecium, or the carpel, beg your pardon, the ovary, we can actually see just about that there are 6 tiny stigmas on top of that ovary, and also that the corrugation or the lumpiness of the ovary itself has got 6 ridges on it. So we have a flower which has got 3 plus 3 tepals, so that would be T3 in this case. We have for the organs that are tepals which have arisen in the position of the sepals, we have 3 anthers opposite those, stamens opposite those, and we also have 3 stamens opposite the petals where the petals will have arisen. But because they all look the same, we're going to call them tepals. So coming to what's called a floral formula, because we're dealing with tepals here, we use the letter T. We put 3 plus 3 because we've got 2 different organ whorls. So these have arisen one set and then the next. We have our andrecium, which is, again, we've got one set and then the next set. So that's also 3 plus 3. And then we have our gynecium, the female organs, which we have said we've got 6 stigmas and then 6 ridges on our ovary. The fact we have 6 stigmas indicates that we have 6 different ovaries fused together. So if there were only 3 stigmas, that would indicate we only had 3 ovaries fused together, or carpels fused together. So here we have a gynecium made up of 6 carpels, and those are fused together. So ideally, and I forgot this earlier, I should have put brackets around that 6 to show that they were joined together rather than loose from each other. So this is our floral formula for the bog asphodel. And this is an artificially coloured electromicrograph of another monocot flower. This is a species, Brumania, which is in a monocot family, monocot clade, which is a group of families called Discoriales. And in this picture, we can see that we have these turquoise coloured organs are our outer tepals. The reddish purpley ones are the inner tepals. These large brown sort of sausagey shaped things are our stamens. So we've got 3 of those. And then in the middle, we have our developing styles. So we obviously have 3 carpels that are fusing together at the base of this flower. In this particular instance, the outer tepals are much bigger than the inner tepals, and that remains the same throughout the development of this flower. And the stamens, there's only 1 ring of stamens being formed here. So the floral formula for this flower would again be T3 plus 3, A3, andrecium 3, and gynecium 3. This sequence of organs forming, in this case it would be outer tepals, then inner tepals, then stamens, then styles. Those organs arise where the concentration of a particular hormone, auxin, is at its lowest on the sort of the lump that is the developing cell bundle, or the primordia, it's also called. So that's an important part of how flowers are formed, and it's something that I will be discussing a little bit more later. So now a little bit on the potted evolution of the evolution of flowers. Our ancestral, this diagram is to show the sort of the characteristics that plants had to go through to, first of all, colonize land, then become something that could stand upright and get off the ground, literally the soil, develop roots so they could anchor themselves and get bigger, protect their reproductive cells, their gametophytes, and then form seeds eventually, develop fleshy fruit, and then flowers and protected seeds and secondary growth. So we're just going to go through this very quickly. So our the ancestor to all plants was somewhere back in time. It led initially to green algae, then brown and red algae, and slowly algae started to colonize the emerging land. Initially as, excuse me, got a frog. Initially as sort of simple liverworts, hornworts, and mosses. To get onto land, they had to be able to regulate water loss, which initially was basically they could desiccate entirely and then be rejuvenated by the application of water. And then in some of the more sort of the later liverworts, that became a waxy cuticle that stopped the release of water into the atmosphere from the cells. Then the evolution of vascular tissue allowed plants to become a little bit bigger. And slowly roots evolved, allowing for the development of things like ferns and club mosses. The gametophyte, so in the case of ferns, club mosses, and everything else before that, the spores that they were producing became bigger, protected, and eventually formed true seeds. And the seeds at some point became covered in fleshy fruit, and that's what you would see with a ginkgo if you were to look at ginkgo seeds. And by the time we get past the gymnosperms, which are cycads, conifers, ginkgos, and gonitophytes, and getting to our flowering plants, monocots and dicots, we have flowers and we have seeds that are fully protected. The main difference between monocots and dicots is the fact that dicots can produce secondary growth. In other words, they can make proper wood, and that's the main, a big difference in between those two groups. So this gives us an idea of the timeline of when all this happened. So we have our sort of very early type plants. We have our Rhynia type plants, which were vascular. They were sitting upright. They were still using spores to propagate themselves, and they had very basic root-like structures, which were mainly for anchoring. So going up through the Paleozoic, we get to things like our seed fern, so seed evolution has happened here. And we see a diversification then into our cycads. And the slow beginnings of moving into what we would know as flowering plants from this point onwards. Conifers were doing very well. Ferns have sort of basically got to their maximum diversification and sort of stayed that way. Same with ginkgos to an extent. And The things like herbaceous lycopods and bryophytes are both fairly stable. The horsetails for equisetae, they had a fantastic time in the Carboniferous, which is where a lot of our coal has come from. But they sort of have reduced their diversity over the millennia. But just make note of the fact that the real expansion of the angiosperms started at this junction between the Jurassic and the Cretaceous. So what was going on in those times? Well, Carboniferous period was 251 million years ago and further back. The Triassic period finished about 200 million years ago, the Jurassic about 146 million years ago, and the Cretaceous about 65 million years ago. And that, of course, is reasonably well known because of the asteroid that hit and caused huge extinction of the larger dinosaurs and the non-feathered dinosaurs in particular. But even so, there were some flowering plants around from 125 million years ago onwards. So things like the Dutchman's pipe, which is Aristolochia, as well as some of the magnolias about 100 million years ago, and the Protea family. And then going into the Cenozoic from this point onwards is when we see the huge diversification of flowering plants. So here we have the reproductive organs of a magnolia. Many spirals of stamens. So these haven't started dehiscing their pollen yet. Excuse me. Many spirals of the female reproductive organs with these large curled stigmas. And they look like they're fused together at the moment in this state, but actually, whenever it comes to maturity, all of these individual carpels will be free from each other and individual. This is across a transverse section through an Aristolochia flower. It's a very strange flower, and there was an awful lot of experimentation with flower shape and form during that period between 146 million and sort of until currently. But in particular, that first 80 to 90 million years, there was a lot of experimentation within the flowering plants for the different types of flowers they might utilize. So we have our stamens here, our styles and stigmas are up here. And this below this structure is actually the ovary. So in this case, the ovary is actually inferior to the rest of the flower. And we'll come back to that term shortly. And this is black pepper. These are the flowers for black pepper. I hope you can see here that we have two tiny anthers, a large sort of reasonably large globular seed with a little stigma on top of that. And here you can see as it matures, it changes color. The anthers are still there, but they've dehisced entirely. They've got rid of all their pollen now. And the stigma just sits on top of that little ball. And this is both, these Aristolochia and Piper are both in a very ancient order called the Piperales. Magnolia, of course, is in Magnoliales. So we're just going to have a look at where they are. But before we do that, I just want to show you this particular reconstruction of fossils that were found in an area in China, very well known for plant fossils from this particular era. And this fossil, which has been named Archaeofructus, this particular specimen is Archaeofructus sinensis. Sinensis means Chinese or Eastern, is at least 124.6 million years old, possibly a bit older, but that's the very youngest it can be. The mature stalks the stamens have fallen off, We have the maturing carpels up here, which are multi-seeded. And then the younger stems, we can see that the stamens are in pairs, and they, oh, I beg your pardon. They have, they're in pairs, they have a little sort of stalk attaching themselves to the stem, and then our ovaries are further up at the terminal part of the stems. So these are flowers. We have male flowers together and female flowers together, so they might not be hermaphrodite flowers, but they are flowers. And we can see a very basic root system, some of which was captured in this incredibly detailed fossil, which, if you wish to have a look at it, the PDF is free to download at using this citation. So our 66 million year point is our what's called the KT, or the KT junction. It's the Cenozoic is also sometimes known as the Tertiary period, hence KT. But it's called the K-PG, which is for Paleogene. So here we've got it at 66 million years. And this period of time between about 100 and about 50 million years is called the angiosperm terrestrial revolution, which is wonderful. I hadn't heard that particular term before. But it gives us an idea of how quickly the angiosperms in particular diversified up until there was some diversification up until this boundary after where the dinosaurs went extinct. And then after that, we have this huge diversification, which increased enormously in what's called the Quaternary. So in the last sort of 5 to 10 million years. As we saw before, our gymnosperms are fairly, I beg your pardon, are fairly static. Same with the ferns and the bryophytes. They've diversified a bit since that point. Fungi have been around for an incredibly long time on land. In fact, they're believed to be probably one of the earliest colonizers of land, around about the same time as algae, if not before. And interestingly, so were spiders. But don't worry, these were probably tiny mites which fed on the mycelium of the early fungus. But what's interesting is how this diversification in flowering plants is also mirrored by the diversification of insects, which are known to be pollinators of flowering plants, and also herbivores of flowering plants, so things like the grasshoppers and aphids. Beetles have been around for a very long time. They started diversifying whenever the first flowering plants emerged, because things like magnolias and water lilies actually heat the area around their reproductive organs, and the petals or the tepals would also fold in over those reproductive organs to protect them during the night. And beetles, unsurprisingly, which like to eat pollen, were very happy to be somewhere warm and protected overnight, protected from predators themselves. So you have this massive diversification of the beetle families. We see the diversification, a lot of the Lepidoptera, the moths and butterflies. Early moths would have been pollinating as well. They had chewing mouthparts rather than sucking mouthparts. And butterflies are a relatively recent evolutionary sort of move away from moths. Flies as well, they've become, they've evolved in that sort of diversified massively in that time period. And of course, our bees as well. One of the things that is an important part of the structure within a plant is how the organs are relative to each other. I've already touched on that to an extent with looking at the primordia, that artificially coloured primordia of the monocot flower previously. And the organs arrange themselves along what happens to be, which is something that has come to be called the golden ratio, which is basically an indeterminate number, indeterminate spirals. So individual spirals out from the center of whatever, but with no, doesn't stop at a particular number. It just keeps going until it runs out of either the gene expression changes or the room. There isn't any room left on the flower of primordia. So here you can see one spiral mapped out, and this is how the golden ratio spiral looks, and that's mapped out using Fibonacci's numbers, which are basically the sum, in sequence, it's the sum of the previous two numbers. So the first number is 1, so the next number is 1. The sum of those two is 2. The sum of these is 3. The sum of those is 5, etc. And this is how you map out Fibonacci's numbers. And of course, plants haven't evolved to follow Fibonacci numbers. Fibonacci came up with that formula to explain the spirals that he saw in the natural world, like, for example, in these agave leaves, and this wonderful sunflower plant flower. But if you have a look closely in the middle of this agave, you'll see that if you count the spirals coming out from it, there are 1, 2, 3, 4, 5. If you look closely in the middle, you can actually see there are 5 points of origin for each of those spirals. And what is happening is right in the center there, which we can't see because it's covered by all the developing leaves underneath this point, will be a functioning meristem, which is actually producing leaves, and each leaf will be developing in sequence from each other, because that is the nearest bit, that's the area that has got the least amount of hormone being expressed in it, so that can produce a new leaf. So it's really quite fascinating how these things work. So it's possible, if this is our earliest leaf, then the one furthest away from it is probably this one. The one furthest away from it is that one. The one furthest away from it is that one. And then the one furthest away from that is that one. So it goes in a sort of a crisscross structure. So it's a fascinating representation of how the genes work in the plant. So here we have the angiosperm phylogeny. Everything in sort of highlighted in yellow here are flowering plants. The very first bits from Umbrellis to Piperales are our very early evolving groups. And these are some of the ones that have the weird and wonderful flowers and have tried out lots of different things. And they are still extant. Umbrellis has only got one species to market. And before that, we have our gymnosperms, the ferns, cycads, ginkgos, and pinales and gnatales. Pinales, of course, have pinaceae. And then we have our magnoliales and our piperales here. We're going to have a look at next we're going to have a look at the family Ranunculaceae, which is in Ranunculales, which is right at the bottom of the sort of a basal group to true dicots, so those with 2 seed leaves, dicotyledons. And we can see the monocots branch off before that. So similarly to the scales of a pine cone, The arrangement of reproductive organs in these very early dicots in Ranunculaceae are still in spirals. Our tepals, in the case of this pulsatilla and hellebore, the tepals, the petals, our petals and sepals are still tepals. There is no differentiation between them. You can only tell which one is first because it tends to be slightly bigger, and if it was green, it might have a bit of red sort of tint to it, where it's been exposed to sunlight and produced the pigment anthocyanin to help protect the cells from damage from the sun. In the case of this pulsatilla, we can see there are masses and masses of stamens going around in big whorls, and then in the center, all of these purple filaments, all these purple sort of little threads here, each one of those is an individual stigma leading to an individual ovary, which will have just one seed in it. With our hellebore, however, even though we still have many stamens, we only have 5 styles and stigmas. Each one of those is going to one ovary, but each ovary will have multiple ovules or seeds in it whenever it develops. And by the way, these green structures, these tubular green structures at the bottom here, are what are called nectar leaves, and that's where bees and so on get their nectar from. That's where the nectar is produced. You might be able to see just at the bottom of these petals here on the pulsatilla, where it's slightly shiny, that's where the nectar is produced on pulsatillas, and it's in a similar location for true buttercups. So we can't unfortunately see the nectaries in these true buttercups, but we can see that in this lesser celandine, Ficaria verna, we have more petals than we would normally expect in a buttercup. And actually, if we were able to see the sepals, we would see there were only 3 of them. In lesser celandine, we get this multiplication, this proliferation of petals, usually which is an indication of how old the population of that particular of those lesser celandines are. It tends to be an indicator of ancient woodland, especially if it has many petals. So it's a good thing to look out for and take a note of in the springtime. Again, we have these spirals of lots of anthers and stamens. And then again, in the center, we have these indeterminate numbered spirals of the female reproductive organs, the carpels. Again, a single stigma going to a single ovary with a single seed. And the same is true for the reproductive organs for this greater spearwort, Ranunculaceae. In this case, however, we have 5 distinct sepals, which you can just see 3 of here in this bud. And then we have 5 large, beautiful yellow petals. So now we've moved from having indeterminate whorls of petal-like structures, tepals, to a pretty much settled number of tepals in the pulsatilla and the hellebore. A bit of a glitch in the case of the lesser celandine, to very fixed 5-petal, 5-sepal structures in the true buttercups, even though our reproductive organs are still in what are called indeterminate spirals. Indeterminate meaning that they don't have a fixed number to them. So if you were writing this, you would use the infinity sign to show that it's an indeterminate spiral rather than a fixed number of stamens or carpels. So what happens when genes that are coding for sepals and petals, etc., go slightly wrong? Normally, whenever a flower is forming, there are certain genes which code for sepal formation, and they're called the Madbox genes, or the A, B, C, D, E genes. A and B code for sepals. B and C code for petals. C and D code for stamens, D code for carpels, and E stops everything from keeping going. So whenever we have something like this Rosa chinensis viridiofolia, what has happened here is that the gene for sepals has got stuck on. In other words, whatever mechanism, genetic mechanism or chemical mechanism within the plant that stops one gene from expressing and starts another one has been lost, and the whole primordia is basically just producing sepal after sepal after sepal after sepal. So we haven't got any reproductive organs, and also our stop gene, our E gene, has gone as well. So this will keep on producing sepals in the center for as long as there's primordia to make it. In the case of this snowdrop, this double snowdrop, we have our tepals here. The large white ones are the equivalent of our sepals, and the greenish ones are the equivalent of our petals. Excuse me. But you can see that we have lots and lots of sepal, of those the green tepals, and the occasional white tepal as well, meaning that we have both the sepal and the petal genes are still are working, but we haven't, the flower hasn't been able to move on to the male reproductives and female reproductive organs. And again, the stop gene has been lost as well. So again, these will keep on producing either sepal-like tepals or petal-like tepals right until the primordia has basically run out of room again. And this slipping of this loss of gene function, or gene function which is a bit fuzzy around the edges, as it can be in some situations, especially in things like the water lilies, you see it, but you would also see it in double roses, is that you get a slippage between the expression of petal formation with stamen formation, and you might see a petal with some a sort of an anther-like structure on the edge of it, which is producing pollen. So it's an interesting thing to look out for if you've got not two double rows that you can pull apart. So our rosaceae, the ones that we've just looked at, and we're going to have a look at more, are further down into our what are called the core eudicots. So basically everything before that would be usually termed basal eudicot, but we don't really need to worry about those particular terms. But I would like you to note that we now have two large groups. We have a grouping called rosids, and we then have a grouping called asterids. Rosids includes everything from the saxifrages right through to the cabbages, and lots of things in between. Malpighiales would be euphorbias, for example. Fabales, of course, is peas. Cucurbitales, cucumbers. Phagales would be beech trees, geraniums. Oh, beg your pardon. Sapindales are things like the maple trees. Acers, malvales are the malvas, the can't remember the name of them. Sorry about that. And brassicales, of course, the cabbages. And then the asterids, we have the caryophyllales, which are pinks, and beg your pardon, the name is gone entirely. The ericas, the gentians, tomatoes and peppers, etc. Borage, the mint family, holly, daisies, and then down here we have apiales, is the celery or carrot family, Apiaceae, And dipsicales are things like teasels. So these are the two large groups within dicots, which are considered to be the most diversified and also the most recently evolved. So let's have a look at some specimens from within the rose family, and also how the receptacle influences go from the point of from this point onwards really affects how the structure of the carpel is in a mature fruit or seed. So here we have strawberry fruit. Sorry, beg your pardon, strawberry flowers. We have our 5 very neat petals. Behind those we have our sepals. We would also have another structure called an epicalyx, but we can't see that, so we can see it here. So the epicalyx here is these aren't sepals, these are another structure which have basically, these are bracts which have changed form and function, so we're not going to worry too much about them. It's a good thing to be aware of in the potentillas and strawberries, that sort of group of the rose family, but otherwise don't worry too much about them just now. We have a multitude of stamens, and we have an indeterminate number of stigmas and carpels. As these sort of are fertilized, we can start beginning to see the development of our ovules, of our seeds. And we can see the ovules are developing quite nicely, but this central bulbous bit that the styles and seeds and ovaries were all sitting on over at this point is beginning to swell. And that is our receptacle swelling. So we can see in this transverse section of a strawberry flower that we have a sort of a large thumb-like structure in the center, which is all receptacle material. And this outer whiter part is the bit that swells. And these are our individual little seeds on the outside, or little ovules, with the individual styles going to each one. As the fruit matures, this whole structure swells and becomes a proliferation of cells, and they fill with, if you're lucky, some sugary substances, so that we get these lovely strawberries. This happens to be an alpine strawberry, which are also very tasty rather than a commercially grown strawberry. But you can see that this hypanthium, the receptacle material, by the way, is called a hypanthium. And the hypanthium swells and matures and colors up and produces sugars, and it becomes red in the hope that it'll attract a bird to come and eat it and carry the seeds away and deposit them with a nice little packet of fertilizer in the same time. We also have other forms of hypanthium. So in the situation of a cherry, we have a cup-shaped hypanthium with all the organs otherwise sitting on the top of the cup, and our cherry forming in the middle. And when it comes to apples, the hypanthium and the receptacle material is actually fused to the outer parts of the carpels, which is what forms the flesh of the apples that we enjoy eating. We can also have inferior ovaries, like in the apple, in other groups. So this is Onagraceae. This is a willow herb. These are fuchsias. We can see we've got 4 very distinct stigmas. Again, 4 stigmas means we have four carpels. And this is the ovary, this fused carpels here underneath the flower. And the same happens with things like fuchsia, which is in the same group. We have our 4 in willow herbs, we have very distinct green sepals and then sort of brightly colored petals. In fuchsia, we have sort of greenish tepals almost, and then we have very small in this particular species, which is a New Zealand species, Excoriatus. We have these tiny little petals, and then we have our four and four stamens, which happen to have this wonderful purple blue, bright blue pollen. So the longer stamens will be the first whorl to emerge, and then the second will be the shorter stamens. And then we have our long style and stigma at the end, which, if we were able to see it properly, would be sort of have four sort of knobbly bits on the end. And here we can see our very distinctive inferior ovary with the fuchsia. Lots of tiny seeds in this structure, and the outer wall of this will become quite fleshy and sweet, again in the hope of getting birds to come and eat it and distribute the seeds for the plant. And this structure here, this sort of large, sort of pillowy bit at the bottom of where the style is, joining the ovary, this is the nectary. So a lot of fuchsias actually recruit birds, especially those in South America. And they tend to be bright red and quite long to use birds as their pollinator. So telling if you have an inferior ovary or superior one could seem quite tricky. If we just sort of flick back a second to our apple, the stem is at one side of the fruit, and then we have our carpels, and on the other side of the fruit we have that sort of fuzzy leafy bit, which is basically what's left of our sepals and our stamens and styles. So if you come to look at an orange, you can distinctly see this not very well, they're not particularly strong sepals, but they are five little sepals attached to the same side as the stem. Again, you can see that nicely here. You've got your sepals underneath the position of the ovary, which tells us that the ovary is above where the sepals, petals, and male reproductive organs, the stamens, are joining the receptacle. So this is a very distinct and very ordinary superior ovary. And then just to confirm it, on the opposite side from the stem of the fruit, we have this scar, which is where our style has come away from the developing ovary. So it's a good idea to look out for where any remains of your sepals are in relation to the stalk of the fruit, should you be looking at fruit. So we've had a look at Rosaceae, we've had a look at Rutaceae, which is the family that oranges are in, and that's in Sapindales, in the same order as things like maples, sycamores, etc. And now we're going to have a look at some flowers in the Lamiales, which are in the mint family, the milkwort family, sorry, not the milkwort, and the Veronica and plantain family. And one of the things that we see, sorry, just going to go back to that a second. We've now moved into our asterids group. And one of the things we see moving into this group, especially as we get further into past the sort of the Caryophyllales, cornales area, sort of into the Ericales and down, we start to see a lot more monosymmetry rather than polysymmetry. So things with one line of symmetry, like a face, rather than things with several lines of symmetry, like a wheel or the base of a bottle, for example. Here we have marsh, a member of the mint family, which is grows in marshes. We can see we've got very monosymmetric flowers. We have a fused, so the flower is fused. It's difficult to tell how many petals you have making up that flower. So a trick is to count the number of sepal points on the fused calyx. So calyx is a collective term for sepals. And if you're not sure whether you've got a mint family or not, have a look inside the fused calyx into this calyx tube and see if you can see the seeds. And the seeds in the mint family are always tiny little sort of seeds. There are only 4 little seeds at the base of this tube. They're called nutlets. And of course, our flowers will be in whorls around the stem, and our stems will be square. We do have a reduction in the reproductive organs in the mint family. We only have 4 stamens rather than 5, and they've all moved to the upper part of the flower, where they're more likely to have contact with any pollinating insects that come to them. This style of flower, and the flower that you also see here in this wonderful ivy leaf toadflax, which is in the Plantagenaceae, and akin to things like the snapdragons. They have a nectar spur at the back, which you can just about see. Actually, you can see one here. The top of the flower is being cut off, and the bees and things have to actually push apart this lower lip from the upper lip to get in and suck up the nectar from that nectary tube. And the snapdragons that people love to have in their gardens, you can often see the biggest queen bumblebees sort of pushing their way into those and having a good drink. Smaller bees, however, will go and actually cut through the flower closer to the nectar and steal it without actually pollinating the plant. So it sometimes backfires a bit for the flowers, especially whenever they're out of their native context. This is a butterwort, one of the parasitic plants that we have, carnivorous plants that we have in the UK. It has whorls of sticky leaves, which little flies and things land on and get stuck, and then the plant will absorb the nutrients from the flies. And they have these wonderful monosymmetric flowers, again with this large nectary tube. And the reason that the flowers are so tall is because they don't want their pollinators to get stuck on their leaves, and not so the flower doesn't get pollinated at all. So you often see this with carnivorous plants, that their flowers are a great distance away from their sticky leaves. And finally, of course, we have orchids, which have very distinctive 3 and 3, but the inner 3 are very different in morphology from the outer 3. The lower one is extremely ornate, and then the upper 2 are very similar to each other, but very different from the third again. We have a reduction in the flowers here as well. We only have 2. We don't have stamens as such. We have the anthers have been placed into this little sac up here, and whenever a bee comes in and goes down this tube, the little sticky points of the pollen sacs stick to either the head of or the back of the bee, and the bee carries them off, and then whenever it gets to another flower that's got a receptive stigma, the pollen sacs are rubbed off onto that stigma. So it's a very effective means of getting their pollinia, their pollen sacs transported from one place to another. And some orchids are a bit mean. They don't have any nectar, although some of them produce oils or for their pollinating insects instead. Another thing that we see in these sort of much later evolving parts of the angiosperms in the asterids is a reduction in flower size, and the number of seeds being produced, and also a flattening of the inflorescence. So here we have an inflorescence that you're all, I would imagine, fairly familiar with, the idea of an umbel. An umbel is basically like an umbrella, but twice. So here it sort of is nice close up here of the individual flowers all coming from the same point on the top of a stem. The same thing happens at the top of the main stem of the flowering part. As you can see, this very flat, lots of tiny flowers is extremely popular with beetles, both types, lots of beetles here, but also with flies, especially hoverflies. And some of the smaller bees as well would enjoy this. Not so much things like butterflies, because there wouldn't be enough nectar available on each of these flowers for it to make it worthwhile them pollinating it. You can see there is some nectar here. This little sort of glossy bit around the flower is our nectar. And each flower has got 5 petals. They frequently don't have any sepals. They have 5 stamens in the position opposite where the sepals would have been. And then in the middle, we have 2 ovaries I've got 2 little styles either side. So this ovary here is made up of 2 individual carpels. You can see that nicely here in this developing fruit from cow parsley. And the stigmas are still there to confirm that we've still got these 2 carpels fused. Once these seeds are mature, they will come apart from each other along that line and fall off the plant or be blown away. Some of them have got mechanisms for that. Some have sort of burrs, which helps them be transported by animals. Oh. So, beg pardon. So our apiales, the carrot family is down here. Apiaceae is in the apiales. That's supposed to read Apiaceae. I'll explain that in a sec. And then we have our daisy family, which is the family that we all we're familiar with, some of them at least, sort of daisies and dandelions, etc. And they have taken the reduction in size of their flowers to the nth degree by producing these huge flat heads. You can see the exposed head of a dandelion with the scars where each of the individual seeds were attached, and through this transverse section as well, the sort of the flat plate that each of the flowers sits on. And you can see nicely here that the central tubular flowers have got these little 5-star petals, and it's a little tubular flower that goes down, and then each of those has a group of stamens, and the style and stigma actually pushes up through that tube of stamens. And in the case of things like dandelions, actually they're self-fertile. They don't need to be pollinated by other dandelions, so they pollinate themselves. But we can also see nicely here these wonderful little 2 curling stigmas. Now, before now, I've been saying that if you've got 2 stigmas, you're going to have 2 carpels. But the daisy family, of course, have decided to do things differently from everybody else. And they only produce 1 fertile seed in 1 carpel, which would suggest that the other carpel is what's called aborted at a very early stage of its development. Either that, or we have 1 stigma. Sorry, we have 1 style, 1 ovary, 1 style, 1 stigma, and it just happens to have 2 branches to it. That is, I realise, something I need to find out for myself. We see this wonderful thistle with all its the white pollen and the styles and stigmas sticking up through those. And the other characteristic that we have, because we have this large plate of tiny flowers, all the bracts that would be normally associated with protecting each individual flower as it develops, instead becomes this huge grouping, especially in the case of thistles, of these bracts creating what's called an involucre. And these bracts are all sort of forming this sort of large cup which protects the young flowers as they're developing. And the same would be true here of this dandelion. And it's not easy to see on this particular one, but there would probably be at least 5, if not more, sort of sepal-like structures protecting the outside of the developing bud. So yes, we have our Asteraceae down here. So that was a fairly brisk run through of some of the weird and wonderful things that have happened to the flowers in their evolution. But just for a few more oddities, just for good measure. Here we have a arum lily, which if I go back to the tree, our arums are this is where I can't find them, of course. Never mind. We have our arum lily, and the spathe, the thing that is sort of it's sort of being held within, is I think bract in origin. At the bottom here we have our ovaries and ovules, and you can see the little thready red stigmas. This red sort of clump here are our stamens and our anthers. And above that, we have this structure which only comes into play whenever the excuse me, the female flowers are receptive. I've finished being receptive, I beg your pardon, and the stamens are about to release their pollen. And the reason they are there is for frequently these arums have a fairly unpleasant smell, which attracts flies. The flies sort of drop down in sort of are attracted by the smell, which is usually coming from this sort of central cup at the bottom. They come down and they sort of, you know, go right happily, sort of dabbing up whatever nectar or whatever the plant is providing for them. And then these fibrous cells expand and close off the entrance stroke exit to this cup-like structure. Any pollen that the fly was carrying from a different plant will have been passed on to the female flowers. Their stigmas sort of die off effectively, and then the anthers, the stamens, release the pollen from their anthers here. So the flies are sat in here overnight, slightly warm, nice and comfy, get covered in pollen, and then in the morning these cells will have withered, and the flies are able to release and go off and fertilize yet another flower of the same species, hopefully. This is a beautiful grass of Parnassia flower, and it is unusual in that, yes, it does have 5, you know, it's got 5 beautiful petals, which has 5 green sepals underneath. We have 5 stamens opposite our sepals, so these are true stamens, and then we have these 5 sort of hand-like structures in front of our petals. And these originally were stamens, but they've changed form and function to help attract in pollinating insects. And these have become nectaries or equivalent. They produce some either a nectar or sort of an oil which is very attractive to whatever pollinating insects are coming to them. And this lovely branch structure is obviously very apparent to a passing pollinating insect. And in the center we have 5 fused carpels as well. So the changes in form and function of flower organs is something that happens fairly regularly and is really interesting to look out for. often it will be a stamen that changes into something like a nectary, but it also can become more petal-like as well. And that's worth looking for. And this is what happens in roses whenever that are being bred to be double. The stamens that would have been produced for the flower are actually sort of become more petal-like, which is why you get that, sometimes you get that transitional petal with anther material on it as well. Last but not least, we have this slightly odd flower from the monocots, the Buxaceae. This is Ruscus, or butcher's broom. You can see it's a monocot because we've got 3 + 3 tepals, and we've got this sort of lovely blobby ovary in the middle and this very shiny, almost translucent stigma. But it looks like it's sitting on a leaf. So a little trick for you is to remember that if there's a flower on what looks like a leaf, then the leaf is not a leaf, it's a stem that just has flattened out and broadened. And often this kind of structure that you will see in plants which are growing in fairly drought-ridden or sort of drought areas which have the potential to be quite dry and droughty. They're not having to produce fragile, thin leaves each year. They can flatten out their stems, which are much more robust and longer lasting. And they can then flower from those stems, as well as having a maximum area in which to photosynthesize and capture nutrients from the sun. Right, well, that is it for this evening. I really hope you've enjoyed the webinar, and if you have any questions, I'd love to hear them.
Thank you so much, Jen. That was absolutely fascinating. And I can see we do have some questions in the Q&A. We've run over a little bit, but if you want to put other questions in the Q&A for Jen, do put them in there. We'll try and cover some of them now, and we may cover others in the notes that go up on the YouTube when we post the recording as well. So first question, Jen, is about if you can recommend any technical books on the macroscopic characteristics of plants. So this is actually linked to your webinar last week as well as this week. So things about the types of leaf edges, types of leaf arrangements, or flower parts.
Mm-hmm. So there are a couple of really good books. So one of the ones I recommended last week, which was Cambridge Illustrated Glossary of Botanical Terms, that would cover a lot about the flower, the leaf edges and inflorescences, etc. There's also a fantastic, I think I put a link in, a link was put into the description of last week's webinar for the Field Study Council laminated sheet, which also covers some of that. But there's also a very recent book, which
Jen's is going to get, and we'll be back with in a second.
This little book is, I hope you can see that reasonably well, is great. It talks about the sort of different structures, so different flower structures. It also covers woody plants, so it will look at the different flowers of woody plants, as well as the inflorescent structures that you see in lots of the common families. It is not a book that I would recommend you use for keying out things, partly because it doesn't have keys, and it doesn't cover all of the species that you would find commonly in the United Kingdom or Britain and Ireland. But it is fantastic if you want to learn more about the sort of the different structural forms of both the vegetative and floral parts of plants, and also the technical terms for those, but explained really nicely and illustrated beautifully. So I would very much recommend that. I will put the link for this book in, I'll have that put into the description for the webinar so you can find it easily.
Brilliant, thank you. The next question is about tepals and sepals or petals. So when a flower has tepals, how does the botanist decide whether they are tepals or sepals or petals?
You can do with care. So usually whenever a plant is if I click through the presentation again and share that quickly, you on the micrograph, can you see that, Julia?
No, you're not sharing your.
Oh, sorry, I beg your pardon. I forgot to press the share bit.
There you are. Yeah.
Yeah. So these outer tepals have arisen in the position of the sepals. So they're usually the first, not the first organs to arise in terms of the primordia, but the first flower organs. Before that, there might have been one bract and two bracteoles. That would be the maximum that there would be otherwise. So in terms of looking at something like a tulip, if you have a look at the underside of the tulip flower, where the stem attaches to the flower, the outer tepals will be holding, so I'm just going to stop sharing that so that I can wave my hands at you all. The outer tepals will be in sort of overlapping the inner ones. So the outer ones will be entirely clear of being overlapped by any other petals, and the inner ones will be overlapped by the outer ones. So the outer ones will be the equivalent of the sepals, and the inner ones are the equivalent of the petals. So it's just through careful observation which ones are at the lowest point, which ones are next up, etc. So you're looking for the ones which are closest to the axis of the sort of the stem of the flower, and then the next ones, the petal ones, will be the ones closest to your male reproductive organs. Hope that helps.
Brilliant, thank you. There is a question here about floral formula, but I think it's going to be quite difficult to do verbally. So I think maybe we could link to some information about floral formula afterwards with the YouTube. But there is a question about nectar leaves. Are nectar leaves neither petals or sepals or tepals?
So nectar leaves are, because they are found on the sort of the tepals are either petals or sepals. Usually, and in the ranunculaceae, in the buttercups, the pulsatillas, the hellebores, the tepals in those are probably sepal in origin. Because they overlap. If you have a look at whenever the wood anemones and things like that come out, they're also in the buttercup family. If you have a look at the back of them, the oldest one will be pigmented because it's been exposed to the sun, and the youngest one will have no pigment on it because it hasn't been exposed to the sun, and all the others will be variation in between. And those are coming in a spiral around, again, in that sort of crisscross section, depending on the expression of hormones. And they, because they are probably more likely to be sepal in function, the nectar leaves are in the ranunculus, the buttercups are on the petals, they've got the nectaries. So the nectar leaves are thought probably to be either stamens which have changed form or function, or a completely novel organ which has then been lost. That's something that is a really good question, and I will do some research and put that onto the description for the webinar.
Thank you, fascinating. And final question, because I'm conscious of time. Can you say again why it's called the KT transition or extinction event? Is K Cretaceous and what's T?
Okay, thanks for the question. So K, yes, stands for Cretaceous, and T would stand for, used to stand for tertiary, which was what the Cenozoic used to be called, so the tertiary period. So it became KT, but now, as I said before, the K still stands because it was the Cretaceous before that event, and it's called it's now called the KPG event horizon, which is PG then stands for the Paleogene part of the Cenozoic.
Wow.
It's probably simpler if you have a look at some geological timescales for that period. That might be more explanatory than me kind of waving my hands around a bit. Sorry about that.
It was fascinating to see the evolution of the flower structure and the way you showcase that. Thank you, Jen. So
welcome.
I'm conscious of time, so we are going to finish on questions there, but we'll put some more information up with the recording. Thank you so much, Jen, for that really fascinating talk. Thank you to Dara for funding our work, and thank you everyone for joining us tonight. It's really good to have you with us. We have another talk on Tuesday next week. We've got butterfly conservation, talking about Lepidoptera and plants. The same time next Tuesday, and then Jen is returning on the 26th of the month on getting started with plant keys. And so you can see this and the rest of the program on the events page of our website. And as I mentioned, the videos will be going up on YouTube. So with that, we're going to bring the evening to a close. Thank you so much, Jen, and thank you everyone for joining us.
Thank you all, and thank you so much for coming. Take care.